다음은 전 세계 100여 개가 넘는 연구실에서 사용되고 있는 콜라겐 접합 물질을 개발하신 분입니다. 유타대 의공학 및 약학과 교수이신 유승주 교수님께서 콜라겐, 신화, 과학, 그리고 바이오 테크놀로지라는 주제로 두 번째 강연을 해주시겠습니다. 여러분의 뜨거운 박수와 함께 무대 위로 모시겠습니다.
So, I think most of you probably have heard of collagen. In Korea, we call them collagen, right? So I do research on collagen, and I've also been lucky enough to develop a technology that's related to collagen. So what I want to do today is to give you some very basic biology, some science behind collagen. I will try to also tell you some story about how our technology was developed. I'll really focus on the aha moment, when the idea came about and how those ideas came about. It really comes from understanding basic science, just like what Steve Rennick has mentioned. I will talk a little bit about some very unique strategy our company is employing in terms of expanding the market. And eventually I will end with my own thoughts about the trends, the direction that biotechnology is moving, and how science can help facilitate that process. First of all, I would really like to thank the organizers for the invitation. You don't really get to talk to this type of general public that often, so it's always a great honor. I would also like to thank, I'll be talking a little bit about the science, so some of the funding agencies from NIH, and also all the lab members who helped produce the data that I will be presenting. Since I'm going to be wearing my academic hat, I do have to mention that I have financial relationship with 3Helix, which makes the material that I'll be talking about today. So I'm standing up here like this, and if you remove everything from my body and just leave collagen, I will be standing up here. I won't be moving. I'll be here. I am not going to collapse because collagen is in your body. It's everywhere. It's in your bone. It's in your tendon. It's in your skin. It's in your cartilage. It's the very fundamental scaffolds of any animals. So the easiest way to think about it is this scaffold that you see, this house. That is what the collagen is. If you look at its structure, the molecular structure of collagens, it's amazingly similar to the construction materials that we actually use every day. So what you see here is a steel cable, which is used to make bridges and buildings. And this is a structure of collagen. It has that fibrous structure. It has a hierarchy of structure. If you go back to the most fundamental structure, which I've shown you at the bottom, it has this triple helix, which is basically a rope. The rope that we use every day to tie things. But what is really, really different between structural material that we use every day and collagen, or almost any biological product, is how we keep them strong and how we keep them for a long time. For structural materials, such as steel cable, we want to make them strong, tough, so that they can last a long time. We can't do that for collagen. We can't really do that for proteins. How do we achieve that? How we make them last longer? Well, we do that by giving it an ability to renew. So collagen gets broken down and it gets made constantly. So we call this remodeling for maintenance. In fact, your bone is going to get remodeled, and there is a lot of, about 20% of collagen is in your bone is going to be remodeled and become a new bone after about 17 years. So the bone that you had 17 years ago is completely different. It's not the bone that you have right now. And that type of remodeling activity is particularly important and seen in collagen. Now, if you have a damage or if you have an injury, you have a situation like this, and we can consider this as an injury or a disease. What do? We usually go and try to fix the house. Of course, we have to do that. But the first thing that you do, even before you're fixing the house, is we usually do the demolition process. We actually have to remove the material before we can rebuild. So degradation or removal of collagen is intimately related to a number of diseases. We're talking about arthritis, cancer, fibrosis, and even aging. So here's a simple situation. I'm showing you a collagen fiber with this is a tendon. You can see its amazing structure. It's made of hundreds and thousands of molecules. They're almost identical. These triple helices, these little rope-like structures. What is really amazing about collagen is that it is a structure, but it is a structure that is ready to degrade at our body temperature. So it's almost like we have evolved our structure so that it can be easily undone, easily degraded. Therefore, if you make a one digestion, if you cleave this protein, it immediately becomes denatured. And when it becomes degraded, it leaves these degraded collagen, and at the same time, it actually produces collagen fragments that can go out to urine and blood. So our technology is a very new way to target and bind to these degraded collagen and also to these degraded fragments that are in our body. So before I move on to some of this work, I do want to touch upon a slightly different topic, maybe for a minute or so, about how collagen is produced, because many are interested in how we can make collagen. Potentially having more collagen can have a lot of health benefits. Well, collagen is protein, right? And the only way to make protein is by linking individual amino acids one by one. So what you're seeing here is a fibroblast, and it does a real good job of making collagen. What it does is that it will bring those amino acids, it will combine them one by one. You have to combine about 1,000 of them to make a collagen. And then it does twist into a rope-like structure, and then they get transported out of the cell. Not randomly, they transport it in a way so that it gets deposited in very well-organized fashion. So if you, let's say, take a collagen product or a collagen supplement, those cannot really be directly used to make collagen. They have to be broken down into small pieces. And those pieces are going to be what supports making this protein. So does that help you in terms of health benefit? It does, because it's a protein, right? Having a protein should help you. But the point is that it is no different than eating a meat or samgyeopsal, right? So if you want to get good skin, go and eat samgyeopsal. All right. So I want to get to the aha moment of our technology. We've been trying to, at least I have been interested in trying to find a way to target these degraded collagen. It would be a situation like this. Well, the idea came from the similarity between collagen triple helix and DNA double helix. There's many similarities, which I won't go into detail, but in DNA, we know a process called PCR. This was just mentioned in our previous talk. In fact, we're using this PCR technology to detect corona these days. And what you see here is that that double helix has to be melted, or they have to be unraveled in order for primer, which is a small DNA, to bind and propagate the PCR process. So the idea that I came up with is exactly the same thing. Can you target these degraded and unraveled collagen strand by using a small collagen-like molecule, we call them collagen hybridizing peptide, and have them hybridize to collagen by formation of these type of triple helix. It's a very simple idea that happened to be somewhat groundbreaking, and you know, I'm really beating my own drum here, but when I first published this work, this idea, I was a newcomer to the field, and many people, expert in collagen, everyone was saying, why didn't I think of that? It's such a simple idea. Well, the reason is people deeply in collagen technology were all thinking about antibody. If you want to find something that can specifically target a protein, most people will say, produce an antibody. You can always find an antibody that binds to something. Well, it turns out that it doesn't work well for collagen, but this idea of hybridization works extremely well. If I look back, and this was probably about 10 years ago when we came up with this idea, and what made this possible. And I really think that this idea came about because of my background in chemistry. I do have a quite unique background. My undergraduate was in chemistry, and then I studied polymer science, which is also heavily science-based. And I was a faculty at Johns Hopkins at the material science department for 12 years, and now I'm doing biology. So having that fundamental understanding of the molecule, combined with the ability to look at the problem not from inside, but from outside, I think that really made possible for me to come up with this new idea. So at this point, I will usually give you the scientific data. I won't really do that. I will just present to you some of the selected publications that we've published. And what this collagen hybridizing peptide was able to do is you can use them, for example, for histology. So if you have cancer, they can take a biopsy, they'll do an analysis, and our peptide can be used just to detect the problem. Very, very simple. It can be used it can be injected into animals, and it will circulate the body. Surprisingly, the peptide stays intact for a long time. It will light up when there is a problem. More recently, and this is published just about a week ago in our latest work, Collagen Biomarkers. In that paper, we were able to use the technology to capture collagen fragments from urine. And I think we can also do this in blood in the future. And by doing more of a big data analysis, here we're showing you proteomics analysis, which can tell us whether you have the disease, more likely whether your disease is getting better because you're taking the drug, or your disease is getting worse because you're not taking the correct drug. And if you are interested in learning more about the technology, you can certainly go to the review papers that I have published in recent years. So where are we going from here? Well, we want to go to the obvious place where we can really help humankind, and that is the diagnostic and therapy. And this is what many people think about when we say biotechnology. And it's really not a big jump from here. All we have to do is, to this collagen hybridizing peptide, we just attach an imaging modality, and we can use to detect disease using CT or even PET. If we attach radioactive molecules, we can attach drug molecules, and not a new drug molecules, but existing drug molecules, anything from small molecule to large molecules, such as antibody. We can conjugate them to our peptide, and that can be targeted toward that specific diseased site. And we can also use them to produce self-assembled nanomedicine. So this technology was originally started when I was at Johns Hopkins. Unfortunately, at that time, it was really difficult to translate that to, or start a company. So I've decided to move my lab where the tech transfer was much more readily available, and the support for that was better, which was at University of Utah. And after I came to University of Utah, and as we decided on to start a company, we really looked at our technology honestly. We could have just gone on and went to the investor, hey, we have the technology, give us the money, let's develop the technology. We really didn't want to do that. I wanted to make sure that we choose the right item. And what we learned was that we have a lot of data in these type of reagent and research, but if we wanted to go to therapeutics and diagnostics, honestly, we weren't really sure how that's, or where we're going to. We didn't even know what disease we need to go after. So we made a decision to attack the top first and then follow up with the bottom. And at that time, we were getting requests from other labs that they wanted to use our material. So what we did was we started a company and we started selling the material. And what we soon realized is that these materials were sold to our researchers all over the world. And it is the researchers that is doing the job that the company wanted to do. They're producing the data. They're telling us where it works. They're telling us which direction the company should go in terms of diagnostic and therapeutics. And this is the best scenario because the data that researchers produce are all peer-reviewed and they're not associated with my company. So what I'm trying to say is that this type of strategy is really not possible for every item. Certain item would be possible, but I also want to emphasize the fact that because the idea came from fundamental science, it's a very breakthrough technology, researchers want to use it and test it in their experiment. And that is what allowed us to push our company into the direction that we're going right now. So these are from the company website, the recent publication that was published. You know, it's used to publish paper in Nature Microbiology, and all very respected journal. It really makes, as a scientist and researchers, happy when you see that what you worked on is now being validated by other research labs. Oops, I think So I want to end with just my personal thought about the direction of biotechnology. If you look at where the technology is going, and we're talking mostly about the biotechnology in terms of materials and drugs and therapy, it's clear that there is a trend. We're going from chemistry of molecules, where we initially think about drug, antibiotics, And then now we are at a stage where a lot of biotechnology is in genes, DNA, and genetic products. So our company could be categorized into that middle. And it is then moving to a more complex system. By combining the molecules and combining the genetic capability, we're now trying to develop engineered cells and engineered organisms. And it is going to happen one way or the other. It may take 5 years, 10 years, but we will start to see these as a real biotechnology products. So it's perfectly fine to follow this trend. I think we should pay attention to what is happening. But what we should not forget and what we should still keep in mind is the basic science that supports these technology. If you think about, let's say, engineered cells, what really made possible is the gene editing technology. So this is a CRISPR technology that was developed, which allows you to edit DNA directly in cells. Just revolutionized the field. That CRISPR technology really did not come from an engineer who tried to make engineered cells. It came from a basic biologist who studied how bacteria's innate immune system, they're trying to protect themselves from virus. So those type of basic science eventually led to this type of application. So I want to end with just two points that from my own experience and from what I have learned, that the basic science is what really allows creation of a real new technology. So Dr. Granick has mentioned about the candle, right? We can always think about advanced form of candle. We can always think about something better. We can always think about a gap in technology. But true gap is a new technology. And it's not easy, right? But I think having a sound basic science gives the potential to create that new technology. And at the same time, if you cannot create the new technology, at least it will help you quickly adapt to that technology by providing manpower who knows the field, by providing instrument and infrastructure who can respond. Thank you very much.